Hi, my name is Cheyenne Olson, and I teach at Northeastern State University. I'm sorry that I couldn't be present today, but I'm actually in class teaching right now, so I'm just going to run through my project, which was measuring crayfish habitat selection and use. I had 4 different learning objectives, and that included number 1, formulating and testing hypotheses about the relationships that we see between habitat features of the stream and crayfish population metrics, such as the number or diversity of crayfish and size of crayfish. We also wanted to collect and analyze data on water quality, substrate, vegetation, and crayfish populations to look at them all together. Apply the scientific method for crayfish species identification, measurement, and population assessment, and then reflect on the influence that any kind of change in habitat characteristics would have on either crayfish distribution, population metrics, or size, and draw some evidence-based conclusions based on what we saw in the field. So for the methods of this, I started with a short introductory lecture on crayfish biology and ecology. And I recommended that for anybody doing this, they also do a short lecture, and in that lecture, try to identify some local crayfish. So for instance, in Oklahoma, we have a scientific paper that came out semi-recently that has all the latest species, and so that's what I used. After that introduction, I actually had the students pick the site that they wanted to look at, and I put in my study that you could potentially do 1 or more sites and compare them. But after they picked the site, they made some observations about the site. We actually went there, they made notes about the habitat, and they began to formulate their hypotheses for what they expected with the crayfish population. We then collected water quality and habitat data, and in this part of my study, I realized it could probably be broken up into 2 different days, where one day is the water quality and habitat day, and the second day is the actual crayfish monitoring measurement day. But we collected data about substrate, vegetation, canopy cover, stuff like that. We then actually collected crayfish, and during our collection, we collected so many we had to stop. There were just too many here. But with our crayfish, we sexed them. We figured out their size of both like chela and total body carapace, and what substrate and or vegetation they were found in. At the very end, I had students in groups, and they came back together to compare what they found based on different habitat or different location, and any water quality metrics that they thought possibly influenced the crayfish. So at the very end of the class, we had a discussion where we just talked about what our original hypotheses were and whether or not what we found in the field supported that. So for me, this was a field day. I managed to do this in about 2 hours with a follow-up lab where we did some data analysis. We just used a local stream that's easily accessible from campus, and I took 14 limnology students with me. Reflecting on this, I actually recommend in my study that you tie this in with other cubes lessons, and so there's a couple on there that do a great job of introducing both like habitat and field sketch, so I recommended those. And I think that this would definitely work better in the future as a 2-day trip where you break up the habitat and the crayfish portion, or you do the field sampling and then the data analysis separate. It just would make it easier for time management. But this also pairs really well with other labs, especially labs you could follow up with, and one that I would recommend is a macroinvertebrate lab. While we were out there collecting crayfish, we collected a lot of macros at the same time. All in all, student feedback said that this was really fun. I had a really good time, so I would definitely do it again, and I appreciate the opportunity. So thanks for having me.